Hello, I'm Nick Ruszala from Bio 200 Lab, and this is my presentation on the Enzyme Lab that we conducted. Alright, so to start off, we assayed enzymes, and enzymes are specific proteins that speed up chemical reactions, and they do so by lowering that reaction's activation energy. And enzymes are important because organisms contain thousands of enzymes that assist many of the metabolic reactions in the body. Without enzymes, many functions in organisms wouldn't even be possible, and we probably wouldn't even be alive without enzymes. The enzyme that we focused on in particular was trypsin. Trypsin is a protease or a proteolytic enzyme that is typically found in the small intestine of humans, and it is also secreted by some forms of bacteria. Trypsin catalyzes the hydrolysis, which is the breaking down of peptide bonds and proteins found in an organism, and this assists that organism in the digestion of many different dietary proteins. In humans, the enzyme can be found in our small intestine, where the pH is around a level of 8. It has been theorized that activity of enzymes it depends on a variety of different factors, such as pH, temperature, and salt concentration, because enzymes are proteins and proteins denature at different levels of pH, temperature, and salinity. To determine the optimum level for enzyme activity for the enzyme trypsin, we assayed its activity by digesting azocasein at varying pH levels between 5 and 11. We thought that the optimum pH level for trypsin activity would be at a level of 9, because that was the result of our last experiment, and it's also close to a level of 8, which is in humans. And we thought it would be 9 because we did not use trypsin from humans, we used it from bacteria. Alright, moving on to the materials and methods. Our substrate was azocasein, and that is a chemically modified protein that was prepared by adding some sulfanilamide groups to the milk protein casein. These sulfanilamide groups absorb light at 440 nanometers, but we'll get back to why that's important in a little bit. Our experiment was conducted in accordance to the laboratory manual by Larry Wimmers and others. Each reaction contained 450 microliters of a buffer, which was at a salt concentration of 15 molar. It contained varying pH levels from 5 to 11. Each different experiment had a different pH level. So one had 5, 6, 7, 8, 9, 10, 11. And then in that same reaction, we had 500 microliters of azocasein. We had 50 microliters of trypsin. And every single reaction, once those materials were combined, they were incubated for 20 minutes at room temperature. After that incubation period, the reaction was stopped by the addition of TCA, which is also known as trichloroacetic acid. And this TCA also precipitated specific macromolecules, including the enzyme and the undigested azocasein from the reaction. Those precipitated molecules were then removed by centrifugation, and that supernatant that we got after centrifuging contained our digested azocasein, and that was placed in a spectrophotometer, and its absorbance was measured at 440 nanometers, because that is the absorbance level of the molecules in azocasein. And since our digested azocasein samples produced a darker red and orange color than the undigested samples, obviously the higher enzyme activity produced higher absorbance values. We also, for our experiment, we had control conditions of room temperature chosen because that was the simplest to keep constant. Our salinity level was given to us by our instructor, because that was just the materials that we were given for the experiment. In our negative control groups, the enzyme was replaced by 50 microliters of water, and this was done to test if our substrate would be digested without the enzyme present. And if it was, obviously, the enzyme really didn't do anything, and our experiment would be false. We also used replicates in order to show that our data was consistent, not just by chance. We had 4 replicates done at each pH level, as well as 4 negative control groups for the pH levels of 5, 9, and 11. So moving on to our results, which can be shown in this bar graph here. Basically, the gist of all our results was that the digestion in the absence of trypsin was found to be significantly lower than that with the enzyme present. So obviously our negative control groups showed little to no activity. Our results also showed that activity increased between pH levels of 5 through 9, and then decreased from levels 10 and 11. So our maximum activity was found to be at the pH level of 9, and there was a significant difference in activity between all varying pH levels between that of pH 9 and pH 10. And to determine that level of significance, we did a variety of t-tests in comparison to the absorbance value that was the highest, which was 9, and we basically tested a null hypothesis that the pH level of the peak was significantly different to each of the levels chosen. In our data, we actually found that the pH levels between 9 and 10 did not have significantly different data, so that was not too great. Our variability between our trials, which was measured using standard deviation, was relatively low, but there were medium to high values for pH levels of 5 and 6. The pH had a 19% standard deviation in comparison to the average, and the pH of 6 had a 17% standard deviation in comparison to the average. But for the rest of them, it was less than 10%. So there was very little variability between between our data. Our enzyme activity was calculated using absorbance readings of the digested azocasein. Our statistical analysis was done using t-tests. Activity levels of 5, 9, and 11 were compared to that of the negative control groups to see if there was any activity. So our activity at varying pH levels was compared to that of the hypothesized maximum of 9. And to define significant difference, we wanted to find a p-value less than 0.05. So overall, we hypothesized that enzyme activity would be affected by a change of pH, as well as that activity would be at its highest at a pH level of 9. Our experiment was adequate to test this hypothesis for the most part. We had the proper number of negative controls and replicates, but our t-test showed a lack of significant difference between two pH values, which proved that we couldn't really fully support our hypothesis. So our data somewhat supported it. It showed that activity would change as you change the pH level. So that was part of our hypothesis, and that was proved. But we could not prove our maximum level of 9 because the pH values between 9 and 10 were not significantly different. But thanks to the results of Group C, which was the only other group to measure pH, they supported our hypothesis, and they found that optimum trypsin activity occurred at a pH level of 9. Our particular choice of control temperature did not allow for our results to show the highest enzyme activity. Groups E, F, and B measured temperature in accordance to activity, and their data showed that peak enzyme activity occurs at 50 degrees Celsius, and we did our test at room temperature, so if we operated at 50 degrees Celsius, we could have actually found the peak activity levels. And we also found that the peak salinity for enzyme activity was at 0 molar, and this was proven by groups in section 8 of this course, which was like another section of the class. So instead of the 15 molar, there should have been a buffer with 0 molar NaCl. Our sources of variability in this experiment are pretty low. The standard deviations were less than 10% of the average for all the values, except for the pH levels of 5 and 6, but they were less than 20, so it really wasn't a very variable activity in the enzymes. The potential sources of variability could have included some pipetting errors, the mistiming of incubation between the samples, we could have labeled something incorrectly, or our pipette could have came in contact with a pellet formed from centrifuging. And between our first and second experiment, there were no major improvements in terms of the replicates and control groups. There were major improvements, like big improvements, because we had no replicates and one control group, so we added a bunch more, and that fully allowed us to test our activity and find the peak. We should have also conducted more experiments at a different range of pH values, including like decimals, to find the actual peak level, as well as operating those experiments at the optimum temperature and salinity levels. Overall, our results suggest that the enzyme trypsin had a direct impact on the digestion of azocasein, and that degree of the effectiveness of its digestion had a direct correlation to the pH level of its surroundings. It would be very helpful for us to measure that activity at a greater range of pH values and at different salinity and temperature values. Our experiment proves that the ideal conditions to grow bacteria are at a specific pH, salinity, and temperature, as bacteria will grow better when its enzymes are more active. For the particular bacteria that we tested the trypsin with, it's most likely going to have its peak activity at a pH of 9 at 50 degrees Celsius and at a salinity of 0 molar. Thank you guys for listening. Hope you all have a nice day.
Dude, it wasn't recording.
It really wasn't.